So the second experiment that we'll be doing is protein digestion experiment, and we'll be using different materials including the Tris HCl of a pH 8.2. And so what we start with is to add 1 ml of Tris to all the 5 tubes. So you have 1 to 5 tubes which are labeled 1P to 5P. So you have to add 1 ml of 8.2 pH buffer solution to each of the tubes. And after you're done with that, then we're supposed to add 1 ml of distilled water to tube 3 and 4. So immediately after adding the deionized water to tube 3 and 4, the next thing you want to do is to put the BAPNA. Remember, the BAPNA is a synthetic protein substrate, and it's made of polypeptides, and we expect that it's going to be digested by the trypsin enzyme from the pancreas or pancreatic juice. So you have BAPNA to all of the tubes except tube 4. So after that, then you take a fresh pipette and you take 1 ml of pancreatic extract and you put that to tube 1 and tube 4. Remember, the pancreatic extract is also known as the pancreatic juice, and it contains a lot of proteolytic enzymes including trypsin, and a lot of them I mentioned in the overview PowerPoint. Then after you're done with that, we are going to add 1 ml of trypsin to tube 5. Remember, trypsin will act as our control, and we are expecting that the trypsin present in the pancreatic juice will be able to digest the BAPNA, and also comparable to what we have in tube 5C or 5P, sorry. So after you finish with that, the next thing you have to do is to vortex to mix the your samples in each of the tubes, and we're going to do that. And after we're done vortexing, we're going to cover the tubes with paraffin to prevent them from spilling over during incubation. And after that, you're going to do the same thing we did for carbohydrate, the ice. You're going to put the tube 2P into the ice pack, and you're going to incubate them for the same period that you're going to incubate the rest of them in 38 degrees centigrade water bath. So that's the water bath, and you put in them there, and we're going to leave them there for a period of incubation, which is going to be for 1 hour.